Welcome everybody to our latest DecodeME webinar. I have to apologise, I'm not particularly well today, so my voice is a bit croaky and hopefully it will last out until the end of the webinar. But I'm really pleased that both Chris and Andy are here with me and know that you'll be in capable hands if I suddenly have to dip out, but hopefully that won't happen. I just want to say welcome to the 500 people who are in the webinar. Over 800 signed up and we're only allowed 500 here. So if you do get kicked out and can't get back in for whatever reason, your Wi-Fi fails, do go to Facebook Live because we are also streaming live there. So welcome to everybody who's on Facebook Live. We've got some slides that we're going to share with you in a moment, but we will also be taking questions at the end of that. Some of you have very kindly sent us questions in advance. I'm afraid we're not able to answer any questions that have got any personal information in. Quite a few people did send those in, so I apologise that you've taken time and energy to do that, but we're not in a position to answer personal things about your own personal health or personal experience. The questions we'll be picking up today are focused on the genetics and on DecodeME. So I have got some questions here that we will try and answer. If you're in the webinar, you can ask questions through the Q&A function. That will be open throughout the webinar. And if you're on Facebook Live, you can post questions there or in our other social media. Colleagues Claire and Anne are both in the background helping set this up, but also monitoring questions, and they will feed those through to me. As always, we will try and get through as many questions as possible, but we never have enough time. So there is a frequently asked questions section on our website, so you can also look there and see if your question's answered there. Okay, so I'm going to start by just outlining what we're going to go through today. We're going to start with a review of 2023. We're almost at the end of the year and we've got lots to celebrate. We're going to tell you a bit about the second questionnaire and Andy will cover that. We're going to talk to you about data access, share some key dates and deadlines, and then we'll move into the Q&A. So I want to start by thanking you all. We're a small team at DecodeME, but there are a lot of people that you don't see that work behind the scenes. They have all worked incredibly hard, have gone above and beyond on many, many occasions and dealt with lots of hurdles. And they all stand with me in thanking you for everything that you've done. Those of you that filled in the questionnaires, those of you that have provided saliva, those of you who've helped promote the study and reach out to others. And together we've created the largest database and resource of ME people in the world. I mean, that is just a phenomenal achievement. We've had over 26,000 people have completed the questionnaire, and that is just a phenomenal achievement. We've also reached more people just by our talking about DecodeME, sharing information, and we're delighted to be moving forward to the next stage with such a strong group of participants. We analysed the questionnaires from the first 17,000 participants and we shared those during a webinar and the results during the webinar, but also we had a paper that was published by NIHR Open Research in August, and that generated substantial national and international media coverage. So not only are we looking at the genetics and the data from the questionnaires, but we are actually raising the profile of ME at an international level. In August, the DecodeME was also recognised in the UK government's ME delivery plan as one of the leading projects in ME research. And we've got a lot of work to do now to analyse the 21,000 samples that we're hoping to receive back in the next phase. So as I mentioned, we closed recruitment on the 15th of November. 26,900 people have signed up and completed the questionnaire, and 21,500 people have been invited to provide a DNA sample. We've sent out 21,600 kits, and so far we've had 17,000 kits returned. So a big plea, and you're going to hear me say this several times throughout the webinar, If you haven't yet had a chance to return your kit, please, please, please do that. We want to get as many kits back as possible, and we know that there are lots of reasons that people have mislaid kits. Dogs have eaten the kits. We've had some brilliant reasons that have come through to us. But if you have a kit at home, please try your hardest to get that back to us. We've already started extracting the DNA, and we've done that with over 15,000 kits already. So we've got DNA samples from nearly 15,000 people. And as I said, the more DNA samples we get, the better our chances of making a statistically significant discovery. So please, please, please help us and get those kits back. So I'm now going to hand over to Andy. He's going to talk to you about our second questionnaire.
Thanks, Sonya. So our second questionnaire, why do we have a second questionnaire? You might be asking yourself. As you can see, and as you've heard, we've already created the world's largest cohort of ME patients. And all of those people who took part will have completed our first questionnaire, answering many useful questions there. But what we wanted to do was ask more questions, but we also wanted to make that first questionnaire as accessible as possible. So we limited the first questionnaire to the essential questions that we needed to ask for that stage. And then we've created this second questionnaire to ask the questions that we additionally wanted to ask, to add more depth, more information to our cohort. So that's basically why. As you can see, our bullet points there, it talks about delving deeper into your experience of your ME, and how your answers will be useful, not just to us when we come to analyze all of the results at the end of the study, but in future research, we anticipate both ME research and research potentially into other conditions as well, who might be interested in using the data that we've collected. Couple of important points that I want to stress. Firstly, that the second questionnaire is completely optional. So if you've completed the first questionnaire, and everybody who did will receive a invitation to the second questionnaire. If you're now at a point where you don't have the resources to be able to complete it, then you absolutely do not need to complete it. It's just, if you like, additional information. And additionally, whether or not you answer the second questionnaire does not affect your previous participation. You will still be participants in the study with your participation already. If you're able to complete the second questionnaire, that would be fantastic, and we would really like you to do that. But equally, if it's not right for you, then basically no worries. We know intimately that ME is a condition that can fluctuate, so we're quite content if you're not able to take part. But equally, we'd really like you to. So if we move on to the next slide. Wonderful, thank you. So just to stress, or just to highlight a couple of dates that relate to the second questionnaire. Now, a week ago, we posted out the paper copies to those participants who have already completed a questionnaire on paper. We did this in order to give those who are most severely affected participants, to give them as much time as possible to complete their questionnaires and get them back to us. As you can see on screen, we'll be sending email invites to our online participants. 10th of January is our aimed for date, I believe. Last I knew, we hadn't settled on that, but perhaps this slide tells me that the team have now been able to decide that it's definitely the 10th. But we're looking at early January for email invites to go out to our online participants. Then you can see the next one, 15th of February, is the deadline to post back paper copies. So those of you who've received a paper copy of the second questionnaire in the post, please get it back into the postal system by that date to give it the best chance to get back to us. And then the deadline for online completion is the 29th of February. So that will be the date that we absolutely have to have everything completed if you want your additional answers to those questions included in our analysis. And as you can see at the bottom, hold on, yeah, thank you. As you can see on the bottom, we estimate it takes about 20 to 25 minutes, similar in line to our first questionnaire. And for those who need it, we again have the 25% group who are kindly providing support, telephone support for those who need that additional bit of help to complete it. And I think I'm now finished. So back over to, well, on to Chris, I believe.
Thank you very much, Andy. So I'm going to tell you about your data. You have entrusted us with your data, and we have not taken your decision to share with us your data for granted. So we take very seriously that trust that you place with us, that we keep your data secure in the University of Edinburgh, in our secure computer systems. Now, we of course want researchers around the world to take advantage of DecodeME data that you have so kindly shared with us. DecodeME data, and that is through your questionnaire responses, and also the DNA data. Now, not everyone has consented to do that, and that's absolutely fine. So what I'm talking today to you about is the set of consented data. We want to make sure that we, as DecodeME, accelerate research across the world so that there is, the first effective treatment of ME as soon as possible, and as many different treatments as possible to apply to as many people across the world. So we want to make sure that there is a robust process by which researchers in academia, in industry, in the United Kingdom or across the world can gain access to the data in a very secure way to ensure that they can take advantage of the data, but without the ability to identify individuals. And that's a key aspect. So what are we doing? Well, we are developing a process that scientists can apply through to allow them to be given the data for consented individuals. Who is making that decision? Well, it's us, Sonya and Andy and myself as a scientist, a patient and charity representative, and we will adhere to the guidelines that we have set up and we stated right at the beginning of this project. We will only share the data that you have shared with us with studies that meet high standards, are ethical and worthwhile. As I said, we will not share personal details that might identify you or any of your health data. Researchers will have to treat your data with the respect that it deserves to keep it secure. Now, we are opening up this process to anyone around the world. And I understand that DecodeME was discussed at the NIH meeting yesterday. So anyone, including in the US, we're going to open up the process in April next year. So is that the last point on that slide? It probably is. I can't see the bottom. So if we can move on. The process that we're opening out is to receive applications from individuals. We ensure that that individual is a bona fide academic or a commercial institution, that what they're wanting to do is rational, it's possible, and it definitely has scientific value. It will have to comply with data security, excuse me, and ethics best practice. We want to know whether PPI is involved, and if not, why not? The funding for it is in place, so it has to be feasible. We are not going to judge the scientific value because in science you can always be surprised. But we want to ensure that it is going to be feasible and potentially has that value that we all wish for people with ME. So there are logistics. We have to ensure that the data that they wish is already available. And we want to make sure that the process of sharing data is practical. There is a contractual element. That often takes time. We'll pass through that to sharing the data. And at the end of projects that use the data will be deleted or returned to us. And if they provided added value to the data, then we'll ask for that value to be passed back to DecodeME to allow others in the future to take advantage of it. So let's go on to the next slide, which I think talks about what our pilot. So we have 2 pilot projects to pilot this process. The first of these was from the University of Glasgow. And this project is all about human herpes virus 6, not the type that you can be infected with, but a type that people have, a minority of people, but some people have, that is already integrated in your genome and has been passed down to you by your parents. So it's integrated. And so there is some prospect that these viruses that are passed down the generations might be reactivated and trigger ME. But absolutely, we need the power of the types of large studies like DecodeME to confirm any association with the integrated form of the virus in ICI HHV6. So their proposal that they passed to us was whether they could determine the frequency of this integrated form of the virus in people with ME versus the general population. And it's the general population data they already have. So if it is found to be more prevalent in people with ME, and perhaps with an infectious onset, then this might help them to understand the biology of ME. So luckily we recognized that there were some leftover samples from the DNA extraction process from the first batch. And we already know that this is a good batch, that the data from it is excellent. And so with that leftover DNA sample, we took advantage of that. We considered their application and realized that what they needed were the questionnaire answers about the infectious onset and the birthplace information, because there is a variation in the amount of this integrated form of the virus as to whether where you come from in the country. So we will, under secure conditions, pass that information over, and the DNA has already arrived in Glasgow, and they are already starting processing the DNA samples. Because, as I said at the beginning, our principle is we want to accelerate research, not by us necessarily, but by people around the world who can add value to what we are already doing. So the second project is different from that. It's a machine learning project, not with academics, but with people in industry. This is Precision Life, who are based in Oxfordshire. So you may know that they have a proprietary combinatorial genetic analysis method, and they have published their thoughts on genetic associations in both ME and long COVID this year. So they have their own view as to how they can analyze the data differently from DecodeME, and we would be very interested in DecodeME to know whether they are able not just to find associations, but whether those associations can be found in different people. So if they analyze 2,000 people and they find things, they analyze another 2,000 people who are different, they find the same things, then there is greater likelihood that these are true and important findings. So they want to extend what they've previously done with analyzing UK biobank data, extend it using DecodeME data to look for these combinatorial risk scores. So are there different genes that when combined, the genetic variants combined, can predict people with ME, and different types of ME too. And that's an interesting aspect to what they wish to do. So we're in the contractual stages of that, and they wish to have questionnaire answers, and they'll need the DNA data from consented individuals only, clearly. Excellent. So I think I've reached the end of what I need to say, and I'll pass you back to Sonya.
Thank you, Chris, and thank you, Andy, and also thank you to everybody who's sending questions through. Just a reminder, we can't answer personal questions. We will be focusing on the genetics and the DecodeME study, and if possible, try and keep your questions succinct because it makes it easier for me to ask them and we can get more in. So just to finish off before we move to the questions, we just want to highlight some key dates and deadlines for you. So the 7th of December, the paper invites were sent for the second questionnaire. Between the 18th of December and the 7th of January, we're actually going to have a complete holiday shutdown for the Christmas period and the New Year. The team have worked so hard. It's really important that they take a break. So if you do send questions in, please be aware that you won't get responses during that time. And also we might have a bit of a backlog when the team come back into the office. So to start the new year on the 10th of January, we'll start sending email invites for the second questionnaire. And just a reminder that we do have the 25% group for providing support. And I have to say that in our first round of questionnaires, they supported 750 people with very severe ME to be able to participate in the study. And those are individuals who didn't have members of family or their support network available to help them. I think that's a phenomenal achievement. Just want to say a huge thank you to Helen, who supported that work from the 25% group. We're really committed to making our study as accessible as possible. By the 31st of January, if you've got a spit kit, please, 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 please return it. We're desperate for you to get that back to us so that we can analyse your DNA. But just a reminder that all of you that completed the questionnaire, whether you've been invited to submit a DNA sample or not, you are still a participant in the study and you still have the opportunity to participate in the second questionnaire. And then on the 15th of February, we're asking people to make sure that they've posted back all their copies of the second questionnaire, the paper copies. And on the 29th of February at 5 p.m, we will expect everybody that wants to have completed their online second questionnaire. So some key dates there for you. So I'm now going to stop sharing the slides, and you will see Andy and Chris to join me for the Q&As. So I'm trying to manage this through the Q&A function. We've got questions coming in from social media, and I've also got questions on my paper. So if I'm looking around and not looking directly at the camera, I do apologise. That's because we're juggling all these different things. So I'm going to start by asking you a question, Chris. It kind of brings together some of the questions that we've had. One on the chat said, will my children get ME? We can't answer that question. We have no way of knowing. But Chris, I'd like you to tell us a little bit about what we do know genetically about the inheritability of ME.
So we do know that there is a genetic component to ME. And the reason we know that is not us, it's previous work by others, that if you have ME, then there's twice the usual likelihood that your second and third degree relatives also have ME. But what I have to say is that this is not definitive. So if you have ME, then it is not 100% sure that your relative is going to have ME. So that means that there is not an inheritance pattern which says with 100% certainty that there is that your offspring and your relatives are going to also have this awful disease. If it were to be so, then it would be something called a Mendelian disease. And that often occurs in single genes, and you can pinpoint where the genetic issue is, and that would be really lend itself to a genetic test, and it would be then rolled out across the National Health Service. Unfortunately, it's not as simple as that in ME. There are lots of different places around the genome that will together contribute to risk. And it will, one place may tilt the balance just a little bit, and another place tilt the balance perhaps in the same way or the opposite way. And so it's quite a complex mixture. And that is why we believe it to be a complex genetic disorder. Rolled up in that is obviously the environment, makes it even more complex. The greatest contributor that we already know about is the fact that people report, 2/3 of people with ME report an infection just prior to onset of their first symptoms. So we know that aspect of the environment is very important, but we also know that genetics plays a part, but not individual genes. So that means it's not predictable.
Thank you, Chris. I'll take a question here. Do you think with the DecodeME study, government will finally take notice of this serious disease? I think, we mentioned that Decode was referenced in the government's delivery plan. I think that there is definitely, the more genetic research that we have, the more that we find, then not just the government, but others will take ME seriously. And as Chris said, we're doing everything that we can to accelerate the research. Both Chris and I were with a minister yesterday, and their interest is there. We have a government delivery plan, but the key to moving things forwards is accelerating the research. We've got questions about results. We do not have any genetic results that we can share with you at the moment. We want to do all that we can to get the results as quickly as possible, but we haven't had all the kits back yet. And as soon as we're in a position to share results that we have confidence in, we will do that. So we really, really appreciate your patience around that. Chris, we've got quite a few questions around ME and pregnancy. Is there anything that you want to share with us that we know from research and that might respond to some of those questions?
Thank you. So I'll deal with the question about pregnancy, but I also want to make the point that it is incredibly important to receive the kits back. And what it isn't easy to do and quick to do after that is to read out the DNA. And we've also got questions from people over the phone and emails, etc., about how quickly the results will come. The process of reading out the DNA by an external company actually takes months. And so it isn't the case that immediately the DNA is extracted, we can just read it out and add it to our data files and do the analysis. It's a bit more, it takes longer than that. So back to the main question here about pregnancy. We know that there are many places across our chromosomes that predispose people to pregnancy issues through genetic factors. So this is a really interesting area. We know, in our study, 84% of people in our study are female. And so that sex bias means that there could easily be many issues that overlap with sex-biased conditions, pregnancy and issues to do with hormonal imbalance. And if we look at the genetic signals from those imbalances and from pregnancy issues, and we look at the genetic signals that come from our study in ME, and if we see that they overlap, then that would provide evidence that there is indeed a biological explanation that covers both. And that's why I think genetics is really important, not just because it explains things in a causal way, but we're able to cross-reference what we find with ME with actually anything that has a genetic signal, and make use of the fantastic advances and discoveries that have been made across genetics for people with ME, to, we keep on saying it, but it's absolutely true, to accelerate towards new therapies.
Thank you, Chris. We've got several questions about whether we're going to widen DecodeME to other illness disease areas, and also whether we're going to open up to other countries. We are funded to undertake the DecodeME study in the UK. We do not have any other funding to either replicate this study elsewhere or to look into other illnesses. As you will know, we did include for those people who had a diagnosis of ME following COVID infection. So we do have some people involved in our study, but recruitment is now closed. We have no further funding to do any more research at this stage, but we will always work with researchers and have been having discussions with others who might be interested in replicating DecodeME elsewhere, and we will share all our knowledge and understanding and the process of how we've delivered this project and support people where possible. We've got several questions around data protection as well, and concerns that the government might be able to access people's data, and also concerns about data protection legislation changing. Chris gave a very thorough kind of outline of how we keep your data safe and how we ensure that everything is anonymized in the presentation. Chris, is there anything you want to add to that as a reminder?
We just take this really seriously, and no, we don't provide a link between your identity, your name, where you're from, etc., your age, with your birth date. We do provide age, but not your birth date. We don't provide identifiable information to others. And that is for this reason. We also don't want any other agency to make the link between the answers that you've entrusted us with, your DNA data that you've trusted us with any other agency. So we break the link, and that is with great purpose, and to ensure that you feel safe in the ways that we're handling and storing your data, and sharing it without that ability of others to identify who you are.
Thank you, Chris. Sorry, another question for you, Chris. This is about the statistical power. What happens if we don't get enough kits back?
Well, everyone so far has just been absolutely fabulous. And the rates of participation, of engagement and talking to us, and the rates by which kits have come back, it's just been fantastic. We're at the sort of 80% level. So I would understand if people can't return their kits. And if that is the case, then we will have less ability to identify the genetic signals that underlie ME. But, there will be good reasons, and I'll understand that. It's just that the more that we get back, the more likely it is that we're able to make these discoveries that should matter in the future.
And just sticking with the spit kits, Chris, I want to send my spit kit back soon. If it arrives over the break, will it be usable? Should I wait to post to you until after we come back from our Christmas break?
It's really stable. So provide your sample. And then it's really stable. It can be posted back as whenever you can.
And just in terms of the results, Chris, can a gene or blood biomarker be established from this study?
So a single marker, no, because it's related to what I said about a single gene being explanatory of ME. Because it will be that there'll be lots of places in our genome that together will be explanatory. It won't be a single gene marker. And so it's not easy. It won't be possible to easily have a single genetic test. Now, that's not to say in the future that won't be possible. But we do not say that this is going to be an immediate outcome of DecodeME.
Thanks. And can you we've got lots of questions. I'm just reading through about what the results may or may not show. So rather than take all the individual questions, I think, Chris, can you just remind us what we are hoping to find, and also a reminder about the potential outcomes, of the study, full stop.
So this is research. Doing research means you never know what the outcome is going to be. You do it to the best of your ability, and then you see what the outcome is. The size of the study for the 20,000 or so individuals indicated that on average, this type of study would find 5 places in the genome that would begin to explain people's ME. Now imagine that each of those 5 places all points to the same thing that is going wrong in people with ME. If it's immunology, if it's neurology, so something wrong with the nervous system, or mitochondria. If everything points to one thing, then that's where future scientific activity should focus. That would be a key outcome. Now, science is very messy, and there is heterogeneity, as people know, diversity of ME symptoms. So it could easily be that those 5 things point in different directions. So one to mitochondria, another to the immune system, for example. It may be that 1 or 2 of them we can't even explain in those terms. So then those individual lines of inquiry should be followed up as soon as possible by the experts around the world, so that their experience and background and expertise and technologies can be brought to bear upon these different lines of inquiry that DecodeME has uncovered for you using your data.
Thank you, Chris. And Andy, you can maybe answer this one. In relation to the data we're collecting and then the access that we're giving, are we going to be charging applicants for it?
I was starting to feel like a spare wheel there, but, for understandable reasons. In relation to the question, no, we're not charging, is the simple answer. Obviously, I can't even think why we would charge, but yeah, we're not set up to charge, and quite frankly, I would be completely against the idea of us charging anyway, because obviously that would be an additional barrier to the value of DecodeME, of the value of all the effort that the DecodeME team is going to, and you as participants are going to, in expending your energy in filling in our questionnaires and sending it back your spigots. So that's a longer answer, but in short, no, we aren't.
Great, thank you, Andy. And I'm going to give you another question now, just that you definitely don't feel like a spare wheel. Have we got any plans to undertake any other questionnaires after the second one?
Simply, again, no. It essentially goes back to funding. During the creation process for our second questionnaire, we still had many more questions than we eventually settled on as forming the second questionnaire. So there would be the potential for us of asking many other things from you, the participants, but there obviously has to be a limit on what we can do given how much funding we have and how much time we have to do it all in. So no, the second questionnaire will be it from us, but obviously you are all you participants are assembled as a cohort. So we are hopeful as well of having researchers coming to us requesting the data, genetic and questionnaire data that was explained earlier, but we're also open to researchers coming to us and saying that they would like to contact those participants who have consented to being recontacted. So again, that's another long answer for what was quite a simple question. But from us, no, there is no further questionnaires after the second one. But hopefully in the future, we might be able to connect those people who have consented with other researchers who want to find out more about you.
Thank you, Andy. And Chris, we've got quite a few questions here. Can you just give us a reminder why not everybody that completed the first questionnaire was invited to then provide a DNA sample, and also why some people that were initially not invited were subsequently invited?
Yeah, thanks for that. So in every study that has ever been put together in genetics, there are criteria for inclusion and exclusion, some of which are, people have to be more than 16 years of age, for example, and that's due to aspects of the law that we needed to adhere to. And there are other aspects, such as we wanted to be reassured that people with ME have a medical diagnosis. And that is so that we can go to others and say, we are in with a firm belief that the cohort that we brought together is one that medical practitioners would recognize as being a cohort of people with ME. So there was always going to be a set of exclusionary criteria and inclusionary criteria. We wanted to make sure that these criteria were internationally acknowledged. So we and this is part of the PPI process, we agreed on the Institute of Medicine, the NAM criteria, and we agreed on the Canadian consensus criteria. So one or both of those. So that means that some people don't come through in our process, which means that we don't ask everyone to provide their DNA. Now, at the very beginning, we wanted to include as many people as possible. We still do, and we set our criteria in a particular way. And we said to everyone at the beginning, we're going to tell everyone at the end of the process what our criteria are. We still do that. But we're not going to say it up front because we feel that others may believe that we are allowing people to change their answers, given so that they would be included or not. So we didn't want that to happen. We don't want criticism of our approach by other scientists, for example. So when we set down our criteria, we were upset, but not as upset as many people who were excluded initially by the reality that not everyone was being invited to give DNA. In fact, relatively few. It was 1 and 2 people at that point. And we hadn't, because this is science, we hadn't appreciated, we didn't know exactly how many people were going to be included and excluded. So then was a sorry for this long answer, but it's very important to explain this, that we then made sure that we, as scientists, we wanted to repair this situation in a robust scientific way. And that's what we did. We went into a scientific huddle for several months and worked out good scientific practices to allow the inclusion of some who had been excluded, but without having any subsequent impact on the quality of the findings that we would then come up with. And we did this by then ensuring that people's comorbidities, etc., can be accounted for in the scientific method. Now, that took time because we needed to think it through, but we also then wrote up a data analysis plan that is online. Everyone can read it. Hopefully, it's accessible so that anyone could understand what we're going to do, how we're going to do it, and why we're going to do it. So I wish that we could turn back time and repair the situation and have invited all of those people at the beginning. But hopefully we've taken the right steps for the right reasons and ensure that as many people as possible could be included in this important study.
Thank you, Chris. And just to give you a break a moment, I'm just going to highlight a few things. We've got quite a few requests coming through. Can we make recommendations? Can we comment on other papers? We're not in a position to do that. That's not what today's webinar is for, I'm afraid. If you have questions, you want access to information, advice, you need some support in accessing healthcare, please contact Action for ME. We have a support line. We've got things on our website. We have healthcare services, but there are also other charities, the ME Association, 25% group, who may also be able to assist. So apologies if I don't take those questions. A reminder that the recording will be available for people afterwards as well. I've got a question here. In addition to the powerful genetic analysis, is there any opportunity to analyse other biomarkers which may vary during periods of remission and relapse? Chris?
If such biomarkers would be accessible in saliva, then saliva samples that are being held as part of the samples you provided would allow such analyses, but they'd need to be present in saliva. And we're told by many people that it's more likely that such biomarkers would be in the blood, which we haven't collected for reasons that hopefully are obvious. It would be far more costly, far more difficult, and people didn't want to do that often. But the recap contact method, so that in future studies that people could be able to donate their blood samples or any other type of biosample, would then enable that kind of study to happen and such biomarkers to be discovered.
Thank you. And I've got a question around consent. Somebody's asked if they can change their consent from no to yes, and how do they go about doing it?
So people have changed their opinion about the study to say that they do not wish to participate any longer, and we withdraw people from the study, and that is absolutely fine. And we said that at the beginning of the study and in the consent form and the participant information sheet. If anyone wants to change their details in any way, please, can you get in touch with us? And on our website, there are contact details of how you are able to stay in touch with us. And we'll see what we can do. Thank you for that question.
Thanks, Chris. We've had a question on Facebook around whether if we wanted to do extra questionnaires, could participants help fund any other work by contributions? It's a really generous suggestion. We've got the funding. We've got £3.3 million to deliver the DecodeME study with the 2 questionnaires that that we've talked about today. So we don't have any plans to do any further studies and further questionnaires at the moment. If you want to make contributions to research, Chris, Andy and I are all working within the Genetics Centre of Excellence that we've set up between Action for ME, Edinburgh University, the ME Association funds research, ME Research UK funds research. There are several charities that you can contribute to if you would like to support further research. I'm just going to quickly go back to the Q&A ones. We've got lots of questions around the links between ME and long COVID, other things like ADHD, fibromyalgia. Chris, can you just tell us a little bit about what we know genetically about links with other illnesses, and particularly around long COVID?
So from the genetics, we don't know about any shared genetic predisposition between ME and any other disease. We know from symptoms that there are shared symptoms sometimes. But the exciting thing from my point of view as a geneticist is this ability to ask the question whether the genetic signals that we hope to see in ME overlap with the genetic signals seen for any other disease or trait or anything, which might therefore provide evidence for commonality, for the same thing contributing to risk for different diseases. So we will do as much as we can on that. And of course, by sharing data, then allow others to do as much as they can on questions that we haven't yet thought about or have not cropped up yet. And that's the reason why we want to share the data as much as possible to ensure that anything that we do is complemented by anything that anyone else could do.
Thank you, Chris. We've got questions. We've got several questions around ME and potential links with ethnicity and increased risk to certain ethnic groups. Can you say again, I'm going to focus purely on genetics, but is there anything that you can say at this point that might respond to those questions?
So we don't know, again, it's early days about understanding the genetics of ME. Now, across the world, 8 billion of us, we share so much of our genetic code that I would be, incredibly surprised if there were to be strong differences between people from different genetic ancestries. I'm not saying it's impossible. I'm just saying to my mind, it's highly unlikely. But what I do think is that it's highly likely that different communities around the world are diagnosed at very different rates. And that is a key thing that I think governments and health systems need to focus on to ensure that everyone has the right access to being diagnosed by well-trained and empathetic professionals.
Thank you. Again, we've got several questions around a viral trigger for ME and what we're expecting to find in our study, but also what's known about whether or not viruses trigger ME. Anything you'd like to say on that, Chris? And Andy, do chip in if there's anything you want to add as well.
So 2/3 of people say that they experienced an infection prior to their first symptoms of ME. So I think that's a very powerful piece of evidence. Obviously, 1/3 have not, and that may be asymptomatic infection or not infection at all. Now, imagine that what we find is that these genetic signals for ME focus on genes that make proteins that are the first point of contact between a virus and our own cells, and that there are genetic differences that change that contact between a virus and our cells. Now, that would immediately shine a light on what is going on and what is going wrong with people with greater risk of ME. we have no evidence for that today, but this is one possible outcome of this type of study. But the beauty of doing a study like this, like DecodeME, which looks across all of the genes, across the whole of the genome, is that we are essentially asking questions of everything. And so all possible outcomes are still on the table.
Anything you want to add, Andy?
I'm not sure I can add anything much to that. I mean, obviously, Chris will be better placed to talk about the science from a patient community point of view. Obviously, the information that we've gathered through the questionnaire, where people have indicated that they did experience an infection before their symptoms started, is really a confirmation of what anecdotally was known in the patient community already. And it's just more evidence to show that that is what is reported by patients. Obviously, it's not cast iron proof of that link, but as Chris says, it is a strong piece of evidence to indicate that infections do often lead, or relatively often lead, to chronic consequences. So they should be taken more seriously than just the short-term consequences that might come from any infection.
Thank you. And please don't put any more questions in the Q&A, because we're not going to have time to take them. So please do conserve your energy. And I'm sure if we were here all day, we would have enough questions to keep us all busy. There are lots of questions around long COVID and ME, and Chris touched on that in terms of a response earlier on. But what we are seeing as a charity is a number of people with ME-like symptoms, with a diagnosis of long COVID, that are needing support. There is research out there that's indicated around 50% of people would meet an ME diagnosis, but have got the label of long COVID. We know it's a very confusing picture for people, doctors included. And so please do go to the charities, and there are long COVID support charities there if you do need some support. Many of us have websites where anybody can access resources there, but people can also contact the charities to gain support if you need it. And that may be in terms of accessing a diagnosis. We know that post-exertional malaise is a key defining symptom of ME. We know that a proportion of people with long COVID have that key defining symptom. So please do access the services available. So I'm going to bring things to a close. I want to thank you all for taking the time and energy to listen, to engage with us through questions, to be part of our study. Even if you didn't complete a questionnaire, many people have reached out to family and friends to promote the study. So you've played a part in lots of ways, and just your support and gratitude. There are so many comments and thanks that have come through on the Q&A, and we will make sure that those are passed on to the wider team as well. We really, really appreciate and value your thanks and your support. There are answers to questions on the decodeme.org.uk website, so please do go there to access more information, and there are blogs as well that you can read. If you want to change your consent, or you have issues around your spit kit that you're wanting to return, anything like that, please do contact us. The details are on the decodeme.org.uk website. We are going to be closed for a couple of weeks over Christmas, but we will play catch up as soon as we can and respond to any contact that we've got. And another thing that was picked out, just as a last point as we finish, there have been several comments that have thanked us for being so collaborative. collaboration is at the heart of this, as is the involvement with people with lived experience. The science is better because of the input that we've had from people with lived experience, either directly through Andy on the management group, through our patient public involvement steering group, but also those that have contributed at early days responding to our consultations and surveys. So thank you to everybody that has played a part now. It's going to play a part in the future, and those scientists that we hope will be able to access your information, your DNA, and hopefully we can accelerate research. Thank you. Goodbye.